Yeah, well, let me just, people come in.
People coming, so
excellent. Yes. So while people are coming in, just welcome to Imaging ONEWORLD. It's wonderful to have people joining us on such a great spring day. Some of us experience the pains of COVID, as I was told is actually our speaker. So we should thank him especially today, because I think, yeah, giving this talk is obviously not painful, but enjoyable. But thank you very much to speaking to us despite feeling hopefully just a little bit unwell. And the speaker today is Krishna Inavalli. He's a senior microscopist, head of the Microscopy Group at the University of Southampton. He is actually a master of acronyms as well. I think he's invented many microscopes which have wonderful, most of them super resolution, I think, techniques, all of them like Sushi, and I mean, just wonderful kind of acronyms. We are very much familiar in the field. He was originally a physicist in Hyderabad and then came to the US, and he was at the University of Illinois to develop novel imaging techniques. And during the second postdoctoral position, actually at the Institute in Bordeaux with the Nagal and Sibarita lab, he developed further cutting-edge microscopes, all of them for super resolution microscopy, and also combined techniques. So we're very curious to hear now, as you are now in Southampton, to hear from the latest inventions and applications. Sorry, I cut it a little bit short, but let me just go and I did a bit of a kind of quick roundup of your career, reading from your CV. So sorry. This was maybe a bit imperfect, but thank you very much, and please show us your kind of work.
Thank you so much, Stefan, and thank you so much, Kirit, for inviting me for this excellent series. Today I talk about the cutting-edge microscopy techniques which have applications in neuroscience. This is a work which I carried during my postdoctoral as a postdoc at Sabrita and John Baptist teams at Bordeaux. So the highlighted region in the brain is called the hippocampus, and this is primarily associated with memories, which are in the form of synapses. And how the synapses forms, that's the communication between two neurons forms the synapses. And if you look at the zoom in view of the synapses, just a comprehensive view of synapse, like all the partners involved in the synapse to form, to maintain, and there are different views of synapses over the time. The first is the classical view of synapse, where free and post spines involved in the synapse and a couple of neurotransmitters. Upon activation, the neuronal transmitters release onto the synaptic cleft, and depend upon the free synaptic receptors, it will form excitatory or the inhibitory synapses. This is the classical view. And for the last 2 decades, people found some other partner play a crucial role in synapses. That's glia cells, that's the astrocytes. And these cells regulate the neurotransmitters. So the people focusing more on the triphosphate synapses. And if you look at the literature last 5 years, that's a lot of papers published on this triphosphate synapses. And given this complex thing, at some point, the molecular exchange between the free post to the glia cells, that all happens to the extracellular space. And this is also important to understand how the extracellular space affect the synapses. So it's a very complex system and a complicated system, and both biologically as well as technically. And the question is that why do we need to unreveal such a complex system? As I said, it's a biological as well as a technically quite challenging. So because Because synapses play a crucial role in memory formation and storage, and a lot of studies shows that the dysfunction leads to the neurodegenerative diseases such as Alzheimer and Parkinson. To give an example, then the Alzheimer is hallmark of the tau tangling and amyloid beta flakes. So it's very important to understand how the tau progress from pre-synapse to post-synapse and the role of different synaptic partners, and at the same time how amyloid beta flakes forms and how they interact with the free and post and the astroglial cells. So these interactions will help us to generate the drugs to treat the Alzheimer's disease at the early stage. So it's given all these factors, it's important to access all the partners involved in the synapses. And to give an idea, like the spatiotemporal organization of these structures, and the synaptic receptors move in microseconds to milliseconds range, and the morphological structures move from milliseconds to seconds range, and those structures are as small as 1 nanometer to a couple of microns. Now, the question comes, can we reveal these structures with a conventional fluorescence microscopy? And the conventional fluorescence microscopy is the widely used technique to image the structure of interest, because it's a EG2 label and EG2 multiplex. You can see 3 or 4 different structures at the same time and compatible with the live cell imaging. Of course, it has a downside that each fluoroscope has a limited photon budget. And these are the 2 widely used fluorescence microscopy techniques, and the wide field microscopy and the confocal microscopy. In the wide field, the emission signal is connected with EMCC and CMOS. And in confocal, the signal is connected with the point detectors, and the pinhole will help us to have the more optical sectioning. However, these 2 techniques have limitations. Those are called diffraction limit and resolution. What is the diffraction limit? The diffraction limit is nothing but the PSF of your microscope, which is close to 200 nanometers. And what it means, basically, the biological structures below the PSF of your microscope looks blur. However, it has a great advantage. If you have a single fluorophore, you can localize with great accuracy and precision. What does it mean biologically? For example, if you have a single receptor, you can know the receptor position with 10 to 20 nanometer precision. So, but in reality, life is much complex, because if you take 1 micron volume, we have plenty of fluorophores. Then the right question to ask is, what is the minimum distance to separate 2 closed fluorophores? This is nothing but the Rayleigh criteria, and which is close to 20 nanometer. What this limitation means? This limitation tells you basically if you have 2 structures and which are less than 200 nanometers, you can't see them as 2 structures. They 2 look like a single structure. So if you want to see 2 different structures as the different, you have to have 200 nanometer distance between them. So what these 2 limitations mean in the neuroscience? So basically the morphological structures are blurred. And if you, for example, if you have 2 spines which are 200 below 200 nanometer separation, they 2 look like a single spine. And what happens with the receptors? Because all the receptors, of course, they're all looks like a cluster. So to overcome these 2 limitations, over the last 2 decades, a lot of techniques come forward. Those techniques are called like super resolution microscopy techniques. And these 2 techniques mostly, there are many ways of implementing crossing the diffraction limit, but the 2 mostly used techniques to achieve resolution below 50 nanometers are called the deterministic techniques, where the stead and resolve falls in this category, and stochastic techniques, where the single molecule localization microscopy like SPT, POM, D-STROM, U-Paint, D-Paint. There's so many names, but all of them have a similar working principle. And there is one more technique, the nonlinear SIM microscopy, but currently I'm focusing on these two orthogonal techniques. So how STED works, this is the conventional confocal microscopy where excitation and emission both are diffraction limited spots. And instead what we do, we go with one more beam that's called the depletion beam in the shape of donut. And this will de-excite all the molecules and emission happens from in the middle. So you have the resolution from this one. The more narrow the null, the more the resolution. And how this works, these techniques, and this is the point scanning technique where the excitation and depletion beams are temporally and spatially aligned, and they scan the whole field of view to generate the final image. And here is an example of 40 nanometer beads imaged in confocal and STED. And you could see the beads are clustered due to their the bead distance is below 200 nanometers. They all look like clusters, but instead they are completely resolved. So how this single molecule techniques works, this is the conventional imaging. What I mean by conventional imaging is that all the fluorophores are excited at the same time. Since these fluorophores distances are below the Rayleigh limit, so these are all looks like the clusters. And in single molecule, people come up with a new fluorophores, and these fluorophores have the different properties, where not all the fluorophores are active at the same time. You can excite two fluorophores at any given time. So as I told you in the previous slide, if you have a single fluorophore, you can see the peak with absolute precision. So what single molecule we do, we excite molecules with a couple of frames. Then once you get the peak information, then we will reconstruct back to get the smiley with unprecedented information, 10 to 20 nanometer resolution. Here is an example to show how the single molecule imaging with the COS7 culture cells with the beta tubing. That's a live. These are the same single molecules, and it's the live reconstruction. This images acquired by Anna Began from the JB's team. So what it basically tells you, each and every point accuracy is lively recorded here. If you combine all these points, you will get the final image with the 10 to 20 nanometer resolution. So what these two microscopy techniques widely used in neuroscience, and STED and resolve used to access the spine morphology, and the single molecule techniques used to access the receptor organization and receptor dynamics and receptor organization. As I said, if you want to have a comprehensive view of synapse, because we want to understand how synapse work and how synapse forms and how they maintain, we need to have both the information, molecular as well as the morphological. So what we need is the correlative SRM with STED and SMLM in one microscope. That will be great, and that will helpful for the biological people to understand the complex synapses. So this is where I started during my postdoc time with the Valentine and JB team. I constructed a microscope where I combined STED with two single molecule microscopy techniques, namely PALM and U-paint. And one is the point scanning technique. As I said, this is the wide field technique. And a couple of issues, because for first thing, we need to make sure we are at the same focal plane, same field of view, because optically single molecule things don't give much freedom, but the optical element STED gives much more freedom in terms of the element. Then I maintained STED focal plane and field of view with respect to the single molecule with respect to SMLM. And another thing is that the emission signal instead pass through different optics and goes to the APD. And in the PALM, the emission signal pass through different optics and goes to the MCC. So end of the day, we want to overlay both the images and talk about the structures, co-localization with 40 to 50 nanometer co-localization. So that is called as the image registration, which is an important task. So I will talk about how we do the image registration to make sure we have good co-localization between STED and SMLM channels. So we use 40 nanometer, 100 nanometers multicolor beads, where we image them in SMLM and STED channels. And once you overlay both the channels, as I said, the signal passed through different optics and different way of imaging them. Then you always see if they overlay in the middle, and you see they are not overlaid in the peripheral parts. This is the well known in the optics community, because even if you use the same channel, same optics, if you do two color imaging, you always see the chromatic shift somewhere. But since it's two different imaging modalities, this is a much more known problem. So how we tackle this problem? We use the coordinate transformation technique. It's a well-known technique in physics community at least. So in that coordinate transformation technique, we take STED as a reference. Then we apply coordinate transformation on the SMLM channel and to overlay both the channels. For example, if you look the green and magenta, green is before coordinate transformation, and magenta after coordinate transformation. Once you apply coordinate transformation, then we have pixel by pixel registration below 40 nanometers. This is very important when you want to implement correlative approaches. Then at the end of the day, you need to see, when you overlay images from the two different modalities, how much pixel to pixel registration you have. So this registration is more than enough for our purpose. Then we looked at the different biological samples to validate the microscope, from fibroblast cells to the growth cones and the matured neurons. And I will talk about the matured neurons, how we access both morphological and molecular information from the synapses. So any correlative approach, the labeling strategy is very important, because you don't want to have much crosstalk between different channels, because the crosstalk will screw your whole method of development. So here we use the dissociated neurons from hippocampus of the embryonic rat brain. And here are the labeling strategy we use. For the STED, we use the soluble GFE or the Sushi calcine. And we look at two important proteins, the PST95 and the AMPA receptor. These two play crucial role in the synapse, excitatory synapses. Then the PST95 tagged with the MUOs, and the GLIA1 we perform in the UPaint method. So how we perform the experiments? First, we take the 18,000 frames in SPT palm. And here is the intensity reconstruction of the single molecules in the palm channel. And we take the three optical sections in the STED, and that's the overlay of all three sections. And then we take then we perform the UPaint, just adding the antibody into the chamber. Then these are the different colors, shows the tracks of glia receptors. So we have very less that time, going from STED to the single molecule, 8 seconds, just changing the port. Once we acquire the palm images, then we go to the STED. Once we get the STED, then we do the UPaint. So here is one example of dendroid spines imaged with the STED, and the synaptic receptor in the palm. We look at the PST95 clusters and the UPaint GLIA1 receptors, and the overlay of all three channels. Then the zoom in shows the all three co-localizations. So thanks to the resolution from all three modalities, STED, palm, and UPaint, and we could able to perform some correlations to correlation of PST95 cluster area with spine morphology, and the correlation of GLIA1 mobility with respect to the spine morphology, and the mobility of GLIA1 in different compartments of the neuron and the synaptic context. So here is the example of different spines showing the PHD95 clusters. And we compartmentalized the spine by looking at the spinules, spine head, neck length, and neck width, and how these parameters correlate with the PHD95 cluster area. So we looked at the neck width and neck length, and the head area. Then we don't see any correlations with the neck width and neck length, and the spine head correlates strongly with the PHD95. What this basically tells you is that the stronger, bigger spine have more PHD95. Then we look at the correlation of GLUA1 with respect to the spine parameters. Because we looked at within the neck length with the mobile immobile ratio of GLUA1, and we saw weak correlation with that. Then we looked at the neck width, the neck length playing role in the GLUA1 receptor movements, but we didn't see anything in that. Then we looked at the spine head, how the spine head relates with the GLUA1. Then we see the strong correlation. That makes sense because the receptors, once they move and they come and organize in the PHD95 area, and they strengthen the synapses. And people showed, I mean, one of our colleagues showed that they form nano domains. So this is expected to see that we have much strong GLUA1 in the spine head. And the bigger the spine heads, as I said, the bigger the PHD95, then the more nanodomains of GLUA1 AMPA receptors. So that's the same thing. We look at the intensity, basically the nanodomains of GLUA1 with the spine head area. Then we saw it's strongly correlated with that. Then we looked at how different compartments affect the diffusion of GLUA1. For example, the synaptic shift, and within the neck length, and within the head area. Head area, neck length and head area. Then once they get inside the PHD95, they confine more. This is what I could expect. They diffuse less compared to other compartments, because this is where the synapses strengthens. Once they get in, they interact with the PHD and they form nanodomains. This is what one could expect them to be less mobile in the PHD95 area. Then thanks to the resolution, to the first time we looked at the spinules. I mean, most of the time I thought if I see something from the spine head, I thought the cultures are not good, there must be something happening. But once we start looking at the literature, even in the live mice, people show that the spines show spinules, and the spinules form the secondary synapses. Then we look at within the spinules how these receptor moves. We found that they move much faster than any other compartment. I mean, these studies need to look in more physiological conditions to understand more on the role of these guys. But this is what we observed, the spinules, within the spinules, the receptors move much faster. So we used the state in the calcine, that's the Sushi labeling. I will explain in the next couple of slides what is Sushi. And this labeling will allow us to see all the cells without labeling them. And we look at the PHD95 and the overlap, both the two showing the PHD95 and the synaptic neurons. And thanks to the PHD95, which is very specific to the post synapse, then we could able to identify the pre and post synapse. So there's the boutons and the spines, synaptic contacts. And we could able to realize the synaptic contact. Then we carried the correlative analysis with the spine area with PHD95 cluster and the bouton area. Then we saw the correlation of both spine head and bouton. So what is the whole study carried in the basic cell cultures, and what are the other partners missing in the comprehensive view of synapse, like the extracellular space and glia cells are missing, and which require tissue or organoid preparation as I said, our motive is to see all the partners in the, which are playing role in the synapse. So in parallel, along with this work, then we focused on another method. We developed in parallel another method, that's the extracellular space, to probe the brain extracellular space. That's called the Sushi technique to probe brain extracellular space. So how the technique works? This is the positively labeled neuron. Just to give you some example, how much information we miss. Most of the time, we go with what we want to see. We label them. But if you ask me from the physicist, you can't alone see one thing because it's all interact. If possible, try to see everything. But unfortunately, that's challenging both biologically and technically. We can't label everything, and at the same time, we can't image everything. So just to give some idea how much information we miss, for example, if you take 1 nanoliter of brain tissue, there are 100 other brain cells and the brain extracellular space. We miss all this information. Just you see only the labeled cell. So what we need is the effective labeling strategy and the super resolved 3D PSL to visualize the everything. So how we gonna see everything? So this is known in the field actually, because rather labeling cells, then label the extracellular space. Just put the dye into the extracellular space. Put the dye into the extracellular space. Then you see all other structures as shadows in the extracellular space, because these dyes are 1 to 2 nanometer size and they occupy all the empty spaces. Then you see all these structures as shadows. So here is an LUT convert. Just I change the lookup table, and all the dark ones looks bright, and all the label ones look dark. So here is one example in the CA1 region. The Sushi reveals different cell types. So you could identify all the morphological different cell types morphologically distinct. But even if you're working long time with astrocytes, or if you're working long time with a particular cell type, then your eyes are trained, you're trained, you could able to see say that, oh, okay, this is the cell type. I could say this might be astrocytes, or this might be pyramidal neuron. So we could identify the different cell types thanks to the unprecedented information. Then with this information, we carried the data analysis with how we carried the data analysis. Okay, the line profile shows the intensity fluctuations. So within the intensity fluctuations, we set the threshold. Then below the threshold, it's all ECS, and above the threshold is the cellular structures. Then the measure of both ECS and the cellular structures. Thanks to the 3D PSF, where we have the isotropy, the resolution in XY up to close to 45 to 50 nanometer. In the Z, we have 159 nanometer. Then we looked at the different regions, how the ECS changes in the different regions of the organotropic slice. Like we saw the changes. Then thanks to this technique, we could able to resolve structures. We could able to see the synaptic clefts and also the wrapping of astrocytes around synapses. And with the 2-color imaging, we could able to find the synaptic contacts with both pre and post synapses, boton and spine, and we could able to perform the data analysis to see the correlation of the boton value with the spine head value. And these are the best things. We could able to see the contextual migration of cells to the first time, since we are revealing everything, whatever moves in that field of view. We could able to see that this guy is pushing everyone in his way. The microglia cell pushing through the brain. Then in another image, we intentionally damaged the we intentionally damaged the slice with the two-photon laser lesion. Basically, I damaged here with the two-photon laser. Then we saw some guys coming and visiting the site, and it will go on bring some more guys to the site. So basically the microglia cells reacting to the laser lesion. Just to sum up, I constructed the correlative microscope where I combined STED with the SMLM, which allow us to visualize both molecular and morphological information. We visualized the KST95 organization with respect to spine, and we looked at the important synaptic protein, how the protein moves within the different compartments of the synapse, and we found that they move much faster than the spinules. And then another technique, we could able to see unprecedented information, all the cells with the Sushi labeling, and we could able to see the synaptic clefts and astrocytes wrapping around the synapses. But however, in this modality, like the STED and SMLM combination, this limited to the basic cell culture due to the SMLM technique limitation. And in this preparation, we missed the ECS and glia cells. And in the Sushi, it provides unprecedented information in tissue and organoid preparation, but however, it's very hard to identify different cell types. It's good to see everything, but at the same time, it's very, very hard to identify the cell types once you zoom in, and the molecular information is missing here. So the best combination to go to visualize the comprehensive view of synapses is that STED plus SMLM plus Sushi plus CI. What that means that we need all these techniques to visualize the comprehensive view of synapse, like all the partners involved in the synapse. What the changes here, STED, then if you go with the 3D parallel structured illumination, then that will allow us this is the wide field illumination and excellent power efficiency, 15 times less power over DSTED and 100 times faster over DSTED. And SMLM, as I said, there's a lot of groups pushing these limitations to take SMLM into the culture preparation. One of them is the selective plane illumination. And selective plane illumination with the multifocal plane detection that will help you to access the 3D information at the same time with the multifocal plane detection. And the Sushi, Sushi is a good labeling strategy to visualize the morphological information, and AI help us to identify different cell types in the so AI helps us to identify the different cell types and enhance the resolution further. So this is the best technique in terms of the next technological development to have a comprehensive view of synapses. So he's Valentin Negron. He's the expert in STED microscopy, and John Baptiste Sabret is expert in SMLM. I was the postdoc bridge between two of them. I surveyed with them 5.5 years. So they're very nice guys. And there's so many people involved in these projects. It's very proud to say that I work with 5-6 different groups to finish this project, the STED SMLM project. So thank you so much. I'm open for the discussion.
Right. Great. What a great talk. Oh, Steph, are you going to do the questions or me? I can do
I think if you I thought you were, but sorry, Rudy.
Right, well, okay. Well, I've got the quiz ready. If anyone fancies doing a quiz, it's all ready to go. If you wouldn't mind just unsharing your screen, and I can get the quiz going.
Yes.
All right, super. It's still there. Okay, got
I
that.
stopped it. Yeah.
Yep.
Gives you a short break as well, Krishna. You're just going to.
Yeah, I think so.
You can have a water and tea.
Okay, let's try and share that guy. All right, we've got a few people in. I love the correlation on Sushi. That I have to say, the imaging there is fantastic. It's very, very interesting to see those other cells going. While we're waiting, I think we've only got a couple of participants, so feel free to sign up. The link is in the chat on the side of the screen underneath. Oh, we've got 6 people. Right. See if we can get a couple more. Oh, 5 people. And then we'll launch the quiz. I think all the answers were in the talk this week, which isn't always the case, but definitely this week they're okay. 7, a couple more, and then we'll launch it.
It always takes a little bit. People need to have a bit of time to get in.
Yeah.
And also, yeah, like put please some questions if you have into the chat so we can pick up on them afterwards.
Yep.
Because that's easier than.
All right.
We have to do it from scratch.
We've got 8 people. Should we go with 8? What do we think? All right. Let's go with 8. So remember, the quicker you are, the more chance you've got of winning, and you get the great fold scope if you're the fastest person to get all the things right. Anyway, I shall launch the quiz now. Here we go. Question 1. Everyone's lining up. And, okay, here we go. So the faster you get, the more points you get. The correlative microscope was developed to access what? Dual color molecular information, or dual color morphological information, or both molecular and morphological information. See if you were paying attention. Yeah, both molecular and morphological. All right, let's see who's on the leaderboard. This is where it gets interesting. Oh, oh wow, Tweeto is there, but it's pretty even. A few people have not got off the starting blocks, but okay, right, next question coming up.
I like we have Carl Gustav from Sweden and the kind of quiz. That's kind of funny.
We have royalty attending. Important technical aspect of developing a correlative super resolution microscopes are matching the focal plane and field of view, that there's no crosstalk between channel. You've got correct pixel registration, or all of them. Let's see what the answer is. And they are all of them, right. Let's see if anything's changed on the leaderboard. Right. Ooh, I think Tweeto is up in the lead again. Let's see, yeah. Well, they're still in contention. It's definitely still in contention. So the quicker you are, the better. Right, there we go. Next question. Memories are stored in the brain in the form of synapses. True or false? Bang. This one should be quick. It's got to be. I think we should have more of these actually, because you can really get speed up on these ones. And the answer is true. Okay. Where are we? Ooh, some more. Oh, the leaderboard might have changed. Yeah, we have a new leader. Puggy is now the leader. Right. Tweeto, you're gonna have to be quick to catch up. And the next question. In which compartment of the neuron do AMPA receptors have less mobility? Is it in the dendrite, the spine neck area, the spine head area, or in the PSD95 area? Definitely saw the answer to this question. Yep. PSD95 area. All right, and the leaderboard. Let's see who's up. Puggy maintained the lead. Oh, it's changed. Oh, mate. Oh, Tweeto, you might be back again. Let's see. Yeah, Pigger has snatched.
Probably guilty. It was probably Tweeto.
Okay, well, I think Puggy Bellens Latan, if you're quick on the trigger, you might get this. Let's see if we can see what happens on the last question.
Doing a very good commentary, like almost like a horse racing.
Well, I gotta make it entertaining. Okay. In Sushi microscopy, how are the samples labeled? By electroporation, in the extracellular space, by transfection, or is it cytosolic labeling? Where is Sushi labeling the sample? Everyone got it.
Yeah.
And that's a great message for you, actually.
Yeah.
Everyone got the point of your talk. Great. All right, let's see who's the winner. Oh, it could be maybe Tweeto's nailed it. Tweeto, if you could identify to us via the chat, the Foldscope will be winging its way to you in shortly. Congratulations. And with that, I will stop sharing. Let's go back to the questions. Ooh, right, so anyway, if you want to write your question into the chat, fine, or if you want to put your hand up and ask, feel free. We're here. Okay. Okay, we have a question from VG. VG, do you want to unmute and ask it yourself? You are welcome to.
Hi, thank you.
Hi, hello.
Thank you. It was a very good talk. Thank you so much. So my question is about dyes. Are there any particular dyes? Because they keep coming up with new dyes, and you probably know of the latest and the best. So are there any STED dyes that you would recommend for the correlative microscopy if we want to combine STED and SMLM?
That depends on, I mean, I don't know at what wavelength you want to do SMLM, because I want to have minimum crosstalk, not at all in that case. So our case, we selected the dyes in the far red for SMLM, and we choose the GFP for the STED. Because most of the time that's the bottleneck to choose the fluorophores when we perform the correlative approaches. So I don't know if you have more, because the STED we are much more limited, to be honest, because only 3 wavelengths you could implement STED, like the 596 and 660 and 773. But the SMLM dyes, you have a bit more freedom to not to overlay both of them. But I don't know, unless I see much more on your spec sheet, then I could say, because you are limited with your lasers, right? It's not about you can use any dye.
Yeah. So would you recommend far red also, because I was thinking in SMLM, you're getting higher resolution. The far red ones are also longer in wavelength, the wavelength itself. Does it matter, or we are not really thinking about those? We are only thinking about dye compatibility with lasers.
I would say when you go with the correlative approaches, it's dye compatibility with the laser. Because the SMLM, even if you push a little bit far, because at the end of the day, I see the accuracy of the PSF. You see that it's you go with 450 excitation or the 700 excitation. Though PSFs are big in the normal diffraction, but in SMLM we always go with the precision. So I don't see much of difference.
Okay. Yeah. Thank you.
Right. Thank you very much. And there's a question from Maria Lima. Do you want to unmute and ask a question if you're still there, Maria? Otherwise, I'll just say it for you. Okay, well, Maria was asking.
Okay, I see that.
Regarding the
nanodomain analysis. Okay, this is the work coming from the same group. It's a Nair before me, and he worked on the formation of nano domains, because people always know PST 95 is a one big area, but then the people slowly realized that it's not everything oxide, but your PST 95 forms nano domains within that region. So that's what I mean by nano domains.
Right. Okay. So do we have any other questions? All right, I had one. So on your channel registration, you're using beads, I think, instead on one hand, and then
yeah, SMLM on the other hand.
So, and what sort of density of basically fiducials do you need in order to get down to about 40 nanometers or better across the whole field of view of your sample? Because presumably you've got to have some reference points everywhere.
Yeah. I think it's fairly good. For example, I did it 40 by 40 micron field of view.
Yep.
And within the 40 by 40 micron, even if you have 50-60 beads, that's fine. But the question comes is that they should distribute within the field, not like clustering in one area more than the other. Yeah.
Okay. And anybody else would like to ask a question?
Hey, Martin is here. Martin Lange. He's the guy who worked on the project before me. The state. Yeah.
Yeah, Martin is actually based in Cambridge, so he's he's gone up. He's hiding. Yeah. Here he is. He's coming back.
Hey. I'm listening in. Yes.
Give us a question, Martin. Come on, don't be
shy. No. I'm not posing a question.
Martin can't ask anything because he's part of the project. So.
Yeah, exactly. So very nice talk, by the way. Very nice presentation.
Thank you so much, Martin.
So, I have a question. So when you were doing the sushi imaging, so here you've really changed the way completely we're looking at an organic typic slice, say.
Yeah.
Were you expecting to see those glial cells migrating around, what are sort of fairly stable dendritic cell structures in the sample?
No, fairly I don't know, because that's the first time we are seeing something moving, pushing every guy to, okay, I want to go somewhere, but I don't know where he want to go.
Yeah, I mean, that's quite a novel observation, certainly from my sphere of complete ignorance of the subject. It really rams home the value of this method. Okay. All right. Are we? I don't think we got any other questions. Anybody else?
I think here the Jan Tonishan, he's the guy for the sushi. Maybe I would appreciate if you put any comment on that question. Jan, are you there?
I saw him before.
He's coming. Yeah.
I don't know if you hear me very well here.
Yeah. Okay.
Yeah, we can hear you.
The internet is a bit off.
Yeah.
Let me move a bit closer to router.
It's fine now, Jan. Well, we could hear you very nice.
We can hear you, so you can just speak anyway without the.
Yeah.
Okay, you hear me now?
We hear you. Yep.
Yeah. So I don't know really. Of course, it was surprising to watch things moving in the slices. Yeah. I can tell the first microglia that I watched moving there, it was a time lapse over, I guess, half an hour.
Yeah.
And I thought that there was drift in the microscope because it's an image every few seconds. So I actually aborted the acquisition and I got annoyed with the slides. And then I just scrolled with the mouse, scrolled through the labs, and then I saw this out of nowhere, the movie, and I was so shocked. And then I, of course, resumed imaging of this dynamic microglia. And I think that I was so amazed by this, and it doesn't show up in the relatively low move speed of the glia sampled by the microscope, but then it's like Christmas Eve when you then get to actually see the time lapse.
Yeah. Wow. Yeah, that's certainly a revelation. Okay. How far have you got with your AI work then, Krishna?
Come again.
How far have you got with your AI sort of implementation?
Yeah, I don't know what's going back in Bordeaux actually, because Jan moved to Spain, I moved to UK, then we two involved in that project. But I see potentially very much with AI, with the Sushi imaging, because that the images, if you look, they are almost look like the EM, but it's alive here. And once you add EM, the AI, then the AI will help us to categorize the cell types. Because that's the hard thing. Seeing is always good, but you don't know what you're seeing is frustrating.
More channels.
Yeah, so definitely for sure. Yeah, really. But I don't know where the JB and the Valentine stands with that part, or even Jan. Then are you trying any AI on the Sushi?
You hear me?
Yeah, we could hear you.
Any AI for analysis, you mean?
Yeah, the artificial intelligence to identify the cell types.
No, not really artificial intelligence. We're doing some relatively primitive analysis of our images, but we are trying to automate it. But I did see there was a very, very interesting preprint from Hans Dansel's lab, I think yesterday or the day before, where they do kind of sushi in fixed tissue. And they also have quite advanced AI analysis and reconstruction. So they aim to do kind of saturated reconstruction of neuropil in fixed tissue. Very interesting.
Okay, great. I think if we don't have any more questions, we may as well wrap up now and let people get on to their lunch. So, Krishna, thank you very much for a very interesting talk.
Thank you very much.
Great movies. And for everybody else,
thank you very much. It was fantastic.
Next week, and there's the title, I can't remember. Oh, Looking Even Deeper, an introduction to 3-photon microscopy.
More deeper.
Yeah. So there we go. Probably the same sort of theme, but just going deeper into your sample. All right. Well, with that, we'll say goodbye to everyone, and thank you very much. And
thank you, Krishna. Goodbye.
Bye-bye.
Thank you very much.
Bye-bye.
Bye-bye.
Hope you're feeling better soon.